We're in Chapter 3, Part 2, Neurotransmitter. Last time we finished the electrical signal of the neuron. So the neurons generate action potential from the axon heel lock. It will go all the way through the axon to the axon terminal. And now we reach the terminal area. And that's the synapse. That's where presynaptic neuron connect with the postsynaptic neuron. And when we zoom in, it looks like this. And there's a small gap called synaptic cleft. It's a nanometer range, 10 to minus 9 meter. And this gap prevents the electrical signal goes from the presynaptic neuron to the postsynaptic neuron. So in this synapse area, the electrical signal need to be transferred into the chemical signal. And that's the neurotransmitter. So this will be our main topic for today. These neurotransmitters, they were packed inside those, we call them vesicle. They are ready to be released. So the terminal area, also called the terminal button. And you have different kind of neurotransmitters. So far, we found 12 different kind of neurotransmitters. And they can be put into two different categories, excitatory and inhibitory. Excitatory neurotransmitter is function as exciting the postsynaptic neuron. How do they do that? They will open the sodium channel. And last time we talked about because of the sodium-potassium pump, the outside has high sodium, inside the cell has low sodium. So when they open the sodium channel, the excitatory neurotransmitter can open the sodium channel. And when they open the sodium channel, sodium going to flow in, and sodium flow in make the inside become more positive, so make it more likely to generate the action potential, and we call this excitatory neurotransmitter. And most of neurotransmitters actually belong to the excitatory category. Then let's look at inhibitory. Inhibitory its function is make this neuron less likely to fire, and it will open chloride channel. Chloride has negative charge. And when the inhibitory neurotransmitter open the chloride channel, when the chloride flow in, inside become more negative and is less likely to fire. So you have both excitatory and inhibitory. Their function are like when you're driving, you need accelerator, you also need brake, and they need to be balanced so your brain can function normally. The neurotransmitter is a big topic, especially for the pharmaceutical companies. A lot of your behavior, your mood, all associated with neurotransmitter, and they need to be balanced. When you have too many, when you have too little, it can cause dysfunction of the person. For example, serotonin is associated with your mood. And people with depression and anxiety tend to have lower serotonin. And 1988 is a big year because they found SSRI, the antidepressant. And SSRI's function is to make serotonin. It will inhibit the reuptake. So after neurotransmitter released, they will be taken back and packed to the vesicle and ready to be released again. So they only stay in the synapse for a short period of time. And SSRI's function is to inhibit the reuptake. So say normal people have 20 serotonin here and people with depression only have 10. Instead of making the 10 become 20, they actually make those 10 serotonin stay here for longer. So inhibit the reuptake. So they can activate the postsynaptic neuron more. And this SSRI's function. So the serotonin will affect your mood. Dopamine is the big neurotransmitter associated with your reward. Like when you eat ice cream, you feel very happy. That's because of dopamine. And when you have too much, it will activate your brain too much. Like when people do drugs, like cocaine or other amphetamine, they will make the dopamine release and activate your neuron like crazy, but they have a serious side effect, which make your system become insensitive to low dosage of dopamine. So you feel not rewarded by your daily activity, like ice cream won't excite you anymore, and also have other effect. And also dopamine is associated with Parkinson's disease. So they affect your life a lot, and that's the neurotransmitter. They affect your daily behavior. And this table tell you, okay, all the neurotransmitter we found so far, so most of them excitatory. So we talk about the dopamine, we talk about the serotonin. Norepinephrine, that's the one used in your autonomic nervous system. Let's look at the GABA. GABA is a neurotransmitter, it's an inhibitory neurotransmitter. So most of them are excitatory. GABA is like the brake you have in your car. So when you drive, most of the time you use your accelerator, but turn out you still need to have your brake, and that's GABA. So GABA is inhibitory. And in a few slides today, we will talk about the tranquilizer. It's a group of medicine. It will directly activate the GABA. Substance P, that's the ones especially used by neuron associated with pain sensation. So when you create the animal, genetically knock out the substance P, they call the knockout mice, and you found their pain tolerance is much higher because they have less substance P, they don't feel much pain. And nitric oxide, there's a gas molecule, there's a less the newest neurotransmitter, because it takes scientists a long time to realize it's a neurotransmitter because it's a gas molecule, and its function in the central nervous system is associated with memory. In the peripheral nervous system, it's a vasodilator. We talk about the Viagra. Viagra pathway it will activate nitric oxide and cause the vasodilation. So these are the neurotransmitters we found so far. And glutamate. Okay, glutamate is one excitatory neurotransmitter. Why I specifically talk about this? Because in our Unit 3, when we talk about the learning and memory, Unit 4, learning and memory, the molecular mechanism of learning and memory, so far scientists believe is in the synapse. So it's not in a neuron. We were born with 100 billions of neurons. Baby have 100 billion neurons. You have 100 billion neurons. But your brain is much more complicated compared with the newborn baby's brain. And it's not the number of neurons, it's the connection. We call the neural network. It happens in the synapse. So every day the baby have new stimulus. It will make their brains, their neuron, gonna extend their axon, and the dendrite form the connection. And the neurotransmitter associated with this is glutamate. And it's a learning and memory neurotransmitter. It's been identified in the brain area called hippocampus. And that's the brain area associated with memory, long-term memory formation. It's not where the memory is stored. So say every day you eat breakfast and you remember your breakfast. And the reason is you have one neuron. They're going to form this synapse connection. And once this is established, and I ask you, what do you eat for breakfast? And you have no problem telling me, oh, I eat Cheerio, I eat cereal, I eat egg, milk. And that's because you retrieve memory from this. And this connection is associated with glutamate. So glutamate need to be released. And it's a very hot topic about 20 years ago. Suddenly they found transgenic mice, smart mice, and they give them more, they have more glutamate. So they can do the maze. We talk about the maze task in the first lecture. And they found they can learn much quicker. And they even try to create the smart pill, so make you smarter by increase the glutamate. It turned out not very successful because it's not that easy. The human's brain is very complicated. Anyway, and this connection is not fixed. It's not fixed. So they can always dissociate and reconnect. So you're always capable of learning new stuff. And after 3 years, I ask you, what did you eat for breakfast 3 years ago? And you will tell me who's going to remember that. And the reason is this connection is dissociated. And it will go to connect with other neuron and form the other memory. And the pros and cons, the pros is you're always capable of learning new stuff. But the cons part is you will forget things. And how to make sure your memory stay there forever? repetition. So in when we talk about the learning and memory, repetition is the key, and repetition will make the synapse release more glutamate and will make it stronger. So this tells your brain, okay, this synapse is pretty serious. I want to remember it forever, and that's glutamate. So your long-term memory, so far study shows your memory is not stored in one brain area. You don't have a hard disk store your long-term memory. It's in the whole brain. It's stored in the synapse. So this is the image fluorescent neurons. This neuron, they put the fluorescent dye inside, and they found the synapse. They're able to grow and connect. So far, your memory is stored in the connection. And this tells you the molecular mechanism, how much we understand about the memory formed. So it's in the connection part, exon terminal part. So at the molecular level, you say some people have good memory, or you study this topic a lot and you memorize a lot of information. And at molecular level in your brain is this part, the exon. And they're gonna have bigger synapse connection, and they are able to put more vesicle there. And every time I ask you this knowledge, okay, you can activate this synapse, and they're gonna release more neurotransmitter, and quickly your memory can be quickly retrieved. So increase synaptic efficiency. That's how we increase your memory. And this part is glutamate's function. Next one, dopamine. So we talk about dopamine associated with your reward. And when we talk about the addiction, we'll talk about dopamine again, and some drugs associated with dopamine. And here, let's look at the Parkinson's disease. Parkinson's disease is the neuronal damage. It happens in the brain area, in the deep brain area. This area called the basal nucleus. The old book called basal ganglia. And this is a group of neurons. And these neurons, they are in the deep part of the brain. They release dopamine. Their function is modulate motor function. So your movement is not one brain area. Your movement starts from your motor cortex. It's in your precentral gyrus. It's in one area in the brain. In our next chapter, next topic, we will talk about the central nervous system. It's in the motor cortex. That's where the signals start. But to make your movement smoothly, you need not just your motor cortex. You also need a lot of different brain area to eliminate unnecessary muscle contraction. Like your cerebellum, we talk about this. This is for balancing. And your basal nuclei, that's the areas eliminate unnecessary muscle contraction. So we compare a good athlete with a beginner. It's not how much power their muscle generate, it's how good they can turn all the muscle power into a smooth movement. And it requires a lot of training, and we call the motor learning. And part of it is the basal ganglia's function. And people with Parkinson's disease, it turned out this brain area, they've been damaged, and they release less dopamine. So it turned out the patient with Parkinson's disease, they have too much unnecessary muscle contraction. So you found they're going to shake their hand a lot of shaking. And eventually they lose their motor function and the language function. That's the Parkinson's disease. So far, no treatment yet. And we could not reverse it. The treatment is give it L-DOPA. L-DOPA is a precursor of dopamine. Like the neuron take to produce dopamine, it take a few steps, and the last step before dopamine is L-DOPA. So we give them L-DOPA, and when the neuron take L-DOPA, they can turn into the dopamine, and it just slow down the process. So far, no treatment of Parkinson's disease. Okay, let's take a short break.